Hello, my name is Styliani Karanika from the Johns Hopkins Center for Tuberculosis Research. In this video, I'll briefly describe a recent study in the Journal of Clinical Investigation on an intranasal therapeutic vaccine designed to help antibiotics cure tuberculosis more effectively by targeting drug-tolerant Mycobacterium tuberculosis persisters. Tuberculosis remains one of the world's leading infectious killers, and treatment still requires many months of multi-drug therapy. One reason treatment is so prolonged is that a subset of bacilli enters a drug-tolerant persistent state that is less susceptible to antibiotic killing. These persisters can contribute to delayed sterilization and relapse, which makes them an attractive target for adjunctive therapeutic vaccination. To address the problem, we focused on REL-MTB, a key regulator of the stringent response in Mycobacterium tuberculosis. REL-MTB senses environmental and biotic stress and promotes a transcriptional program that favors survival rather than growth. Through this pathway, bacilli adopt a low metabolic drug-tolerant phenotype that is much harder to clear with standard drugs alone. That made REL-MTB an appealing target for a host-directed immunotherapy directed specifically at persisters. To target this pathway, we designed a DNA vaccine encoding a MIP3-alpha REL-MTB fusion. MIP3-alpha or CCL20 binds CCR6 and attracts immature dendritic cells. By delivering this construct intranasally directly into the respiratory tract, our goal was to concentrate antigen uptake and presentation in the lung mucosa, where TB bacilli reside, and to generate strong REL-MTB-stimulated T-cell responses at the site of disease. In other words, we wanted a vaccine that not only targets a key persistence pathway, but also sends that antigen straight to the dendritic cells that can best prime protective immunity in the lung. In immunocompetent C57BL6 mice with established TB, we combined intranasal MIP3-alpha REL-MTB vaccination with a standard 4-drug regimen, rifampin, isoniazid, pyrazinamide, and ethambutol. Compared with the 4-drug regimen alone, the combination produced lower lung bacterial burden during the 6-week treatment. Because human TB is characterized by necrotic granulomas, we next tested the approach in C38EB mice, which develop lung lesions that more closely resemble human disease. In this more stringent granulomatous model, intranasal MIP3-alpha REL-MTB again improved treatment efficacy over first-line drugs alone and offered reduced lung inflammation and pathology. The vaccine accelerated sterilization, achieving culture negativity by 12 weeks in all vaccinated animals. Beyond reducing bacterial burden during the treatment, the combination also improved durable cure. 12 weeks after stopping treatment, none of the vaccinated animals relapsed, whereas all control animals receiving the 4-drug regimen alone had detectable bacteria, establishing a stable cure phenotype. We then examined how the vaccine reshapes local and systemic immunity. Using flow cytometry in the vaccine group, we found enhanced and sustained recruitment and activation of dendritic cells together with increased frequencies of REL-MTB-stimulated T-cell producing cytokines associated with TB control. These findings suggest that the vaccine helps the immune system recognize and attack bacilli in the drug-tolerant state that would otherwise survive chemotherapy. We also examined the pulmonary immune response through spatial immunofluorescence. Intranasal MIP3-alpha REL-MTB increased recruitment of dendritic cells in the lung and strengthened crosstalk between dendritic cells and T cells, indicating that the vaccine further reprograms the local immune niche to better recognize and eliminate persisters. Importantly, the benefits of MIP3-alpha REL-MTB vaccine were not limited to the classic first-line therapy. When we combined the vaccine with a regimen designed for drug-resistant TB, we again observed greater reductions in lung bacterial burden compared with antibiotics alone. We next asked whether this platform could elicit comparable responses in a non-human primate species that more closely approximates human immunity. In rhesus macaques, intranasal MIP3-alpha REL-MTB vaccination generated REL-stimulated T-cell responses in both peripheral blood mononuclear cells and bronchoalveolar lavage, mirroring the responses observed in mice. This cross-species immunogenicity supports the translational potential of targeting REL-MTB with a mucosal and dendritic cell-directed vaccine. In summary, intranasal MIP3-alpha REL-MTB vaccination targets a key regulator of the drug-tolerant persistent state, enhances the efficacy of standard TB therapy, reduces relapse, and limits lung damage in preclinical models. By strengthening dendritic cell T-cell interactions and focusing immunity on REL-MTB expressing bacilli, this host-directed immunotherapy offers a promising path toward shorter and more durable TB treatment. Future work will determine how best to advance it toward clinical evaluation. Thank you for watching. Read more in the full article.